Hello, everyone. First of all, I would like to thank you for this opportunity. As introduced, my name is Danilo. I'm from Santos, Brazil. And today I will talk about how we can use experimental designs and simulations to predict treatment outcomes, in this particular case, disturbance and migration's effect on ecological communities. We use nematode assemblages. So this works is one of the chapters from my PhD thesis, which was supervised by Gustavo Fonseca. So I invited him to anytime to make interventions. We get very interesting results, and I hope that everyone can enjoy. So, so I will first briefly introduce how theory, experimentation, and modeling simulations are integrated to produce the scientific understanding. Theory is formulated to explain observed phenomena, whereas experimentation is probably the most objective way to confront ecological theories. It because through experimentations we can formulate and test precise cause-effect hypotheses. The results from experimentations serve as input to new and existing ecological models, which in turn can be used to simulate scenarios, generate prediction, promote new testable hypotheses. We can also ask how well simulations can predict the experiments, which is a good test of whether you have considered all factors relevant to the experiment. Finally, results from modeling evolve theory and feedback into new experiments. Once we have achieved good predictions, we can extend the experimental results beyond the conditions they were primarily generated. So the most effective way to getting good predictions is use experimentation to guide modeling and simulations, and the modeling simulations to guide the design of experiments. So for that, we start with defining an ecosystem schema based on the fundamentals of the experiment. It means that we have to include in this schema all the process with ecological meaning for the experiment. These processes are implemented through mathematical equations. For example, we can have an equation for birth rate, for death rate, immigration, dispersal. So the results of simulations can be compared to the experimental data, and the experimental data can be used to calibrate the simulations. And the use of the observed data to calibrate the simulations, we call this pattern-oriented modeling. So briefly, pattern-oriented modeling is basically in pattern-oriented modeling, we use the observed patterns to restrict our hypothesis about parameters, process. We use the observed patterns during the entire modeling process. We set the initial values using the observed data, and we can recalibrate the model using the observed data until we achieved what we called good predictions. So in this study, we asked if controls can be used to simulate treatment. In other words, can the outcome of treatment, can the parameters extracted from controls be used to simulate outcome of treatments? Can I predict treatments using parameters obtained from an experimental control group? So this was our ask for this work, our question for this work. And to answer these questions, we designed a microcosm experiment which guided the calibration of an individual-based model that I will explain what is later. The microcosm experiment was designed to test the disturbance immigration's effect on nematode assemblages. Well, disturbance and immigration are key factors driving the structure of communities. Disturbance is the major cause of extinction of low-density species, acting as a deterministic driver. Immigration, on the other hand, acts in the opposite direction, preventing the local extinction of species. So we established a very simple hypothesis that disturbance decreases abundance, species richness, evenness under low immigration rates. But if we have high immigration rates, we increase abundance, richness, and evenness. So this was our experimental hypothesis. So the design of was the design of our experiment. The microcosm consisted of glass beakers filled with sediment containing the fauna. Part of the sediment matrix containing the fauna was maintained in a plastic box during the whole experiment to serve as input for immigrants for the experiment. So we have a control group that was sampled after 3 days just to get the initial fauna. We lost one replicate during the sampling process, unfortunately. The other microcosms were sampled after 27 days and included one control group without any disturbance and immigration events and 2 treatments. So the disturbance happened in both treatments. So the disturbance consisted of first here is inverted. So we first homogenized each microcosm and removed about 10 grams of sediment and associated fauna and replaced the sediment by Anazoic sediment. So we count the 10 grams of sediment, and we count that each removal was correspondent to 100 individuals. So immigration events, on the other hand, consisted of adding 100 individuals. So we had 2 treatments, the low immigration treatment, with addition of 100 individuals every 6 days, and the high immigration treatment with addition of 100 individuals every 2 days. So this is the implementation of our individual-based models. So that means that the model is based on the individual. So the model consisted of the cell, which represent the microcosm, homogeneous environment, the species pool, and the individuals. The species pool represents the total number of species. So the total number of species within the cell, the simulated microcosms, is regulated by the parameter carrying capacity. So the model operates by discrete cycles of death, induced mortality. It means that the mortality is like environmental selection, deterministic death. In this kind of death, species identity matters in determining if an individual are going to live or die. We have birth, and we have the immigration in the cell. If the cell exceeds the carrying capacity, individuals are removed until the local population is reduced to K, carrying capacity. So in summary, we perform a search in the parameter space. The parameter space of the simulations include the full factor space of birth, of selection, birth, death rate, carry capacity, and we also explored 5 assumptions of species equivalence. So we call these assumptions of fitness constructions. So in the first fitness construction, we considered equivalence across all species. It means that all species are considered equals in their ecological properties, such as the probability of giving birth and dying. The second fitness construction, consider equivalence within a same CP group. The CP group is the colonizer persister scale. So it ranges from 1 to 5, and being 1 the more opportunistic species and 5 the more persistent species. So we considered all species within a same CP group as equal. So a third fitness construction was considering the fitting type 1A, 1B, 2A, 2B. We also considered constructed fitness based on abundance groups. So the most abundant species were considered equivalent, and so the others abundance groups. And the last fitness construction considered is consider all species as non-equivalent. So it means that all species are different from each other, having unique birth and death rates and response to disturbance. So our simulation starts using the same number of species and the same set of species abundance observed for the control microcosm 0, the initial assemblage. So we start the simulations with these assemblages. We generate more than 2 million of parameterizations. Actually was almost 3 millions. And at this point, we turn off the disturbance and immigration effects in the simulations. So we used a two-step filtering to select these parameterizations. So in the first filtering, we selected the models matching 9 observed univariate index, which we call summary statistics. So we calculate for the final assemblage the number of species, the number of individuals, evenness, an index for dominance, index of trophic diversity, index for diversity, Simpson, Meiolive, index for heredity, maturity index. So we use these 9 summary statistics to filter the parameters. We define the 25 and 75 percentiles of the observed patterns as limit to define a match between simulations and control microcosms. So after we used a multivariate filter, we select parameter sets using a MDS. So we select parameterization that produce communities with the same multivariate structure of the control F. The advantage of using multivariate analysis is, as a second filter, is that it accounts for simultaneous changes occurring in species abundance while preserving species in the identity, and calibrating a model against multiple patterns increases the chance of capturing the internal organization of the system. So we compare the simulate treatments with the observed patterns. In this point, we use these control parameters, and we turn on the disturbance immigration events. The simulate treatments were compared with the observed treatments. So failure in simulating treatments means that we must reject or recalibrate the model. So in the recalibration context, we are interested in tracking slight variations of parameters rather than obtaining a new and accurate model calibration for the available data. So this is our microcosm results. We recorded 42 nematode species where we observed that both abundance species richness decreased in the high immigration treatment, whereas evenness increased. Therefore, our experimental hypothesis was rejected. Actually was the opposite. Our post hoc analysis showed no difference between low and control, entre low immigration treatment and control, so we didn't have this difference. And this is the multivariate, the MDS. These ellipses refers to 95% confidence ellipse. And we also found just difference between high immigration treatment and from both control and low immigration treatment. So this is from more than 2 million parameterizations, only 23 match the univariate patterns, the summary statistics calculated for the control F. This is a redundance analysis where we evaluate the influence of the parameters on these 23 simulations. As response variables, we use the summary statistics calculated for these simulated communities. And we can see that carrying capacity and the fitness constructions were the most relevant parameters influencing these simulated communities. So this is the result for the second filter. So here we place together the communities generated by the 23 simulations and the communities generated by the control simulations. We put a confidence ellipse and we selected the simulations, the parameterizations that were inside the control group. So the colors represent is a formula for measuring the equivalence between species. So 0 means that species are not equivalent at all, and 1 equivalent at all. So the second filter selects only 2 parameterizations, and these both parameterizations predict select on 2 parameterizations. It both predict species equivalence among species within a same CP group. So the CP group was the main factor predicting the communities. So we can make some considerations in natural conditions. Nematodes species within a same CP class may have different niches, resulting from small difference in life history traits, including fecundity, tolerance, environmental stress, and interspecific competition for resource. But our results suggest that under laboratory conditions, these differences are too small, and individuals from different species within the same trait can be considered equals in their ecological properties, such as their probability of giving birth and dying. So this is the result from our simulate treatment. So we turn on we use these 2 parameters, these 2 parameter sets. We turn on immigration disturbance events. This is the MDS result, where the red one, this big ellipse in red is the high immigration treatment observed. It is yellow one is the observed for low immigration treatment. So we predict only the low immigration treatment. So this is the result from the 2 models that we selected. So the green one, the green one represents one model, and the blue one represents the other model, the other parameter set. And both simulations were included in the low immigration treatment. So we predicted only the low immigration treatment. So as I said at the beginning of my presentation, failure in simulating treatments would constitute a rejection of the model or a need for recalibration. So given the importance of K, the carrying capacity towards the control, we recalibrate this parameter using the treatment data. Specifically, we adjusted the carrying capacity of the control parameterization proportionally to the abundance of the observed microcosm treatment. So we take the final abundance of the treatment and adjust just the carrying capacity to match this this parameter, and so we were able to predict the directional change in observed in the microcosm treatments. So I have both low treatments simulated are smaller ones, are both inside the observed, and both high immigration treatments, simulated treatments were inside the observed high immigration treatment. So we also can have some considerations that carrying capacity was the main factor regulating, that was the key factor. So we know that communities regulated by carrying capacity generally show intensive inter-specific competition. So more individuals arrive, the greater is the variability in the use of resource by species, which increases the symmetry of competition and consequently the assemblage evenness. So when immigrants do not successfully reproduce, yet use valuable resource, they effectively reduce the carrying capacity and favor species able to use alternative resource. So in our study, we're calibrating the simulations with the most influential factor in the reduced parameter space was critical to fully predict our experimental results, highlighting the importance of the feedback between empirical and simulated outputs. So I would like to thank you for everyone for this opportunity, for Gustavo, for all the support. I'm sorry about the Rus English, and yeah, I'm open for questions.